Hello students, for today's experiment we are going to dissect the given flower, and to display different whorls we are going to dissect anther and ovary to show number of chamber. So here I had taken a typical flower, that is hibiscus, also called as China rose or jaswant or gudhal. So these are the different name of this flower. So flower is highly condensed and modified shoot for sexual reproduction, which result into seed and fruit after pollination and fertilization. And a typical flower has 4 types of floral leaves called as sepals, petals, stamen and carpel in distinct whorl, that is known as calyx. You can see calyx, corolla, androecium and gynoecium respectively. The sepals and petals do not directly involve in sexual reproduction, therefore they are called non-essential floral organ. While the stamen and carpel are directly involved in sexual reproduction, so they are called as essential whorls. So this is about the introduction of this flower. Now we are going to dissect this hibiscus flower to display their various parts. And for that, here we need certain things. So here these are my requirements, you can see. So for this experiment, for the dissection of flower, I need this needle, forceps, then I need this blade or razor you can take, then I need slide, cover slip. Here I had taken hibiscus flower. This is Hibiscus rosa sinensis or China rose. So these are the requirements for the flower dissection, and now let's start with the procedure. So firstly, you have to obtain the fresh flower of hibiscus, or you can take any other flower like brassica or catharanthus. First of all, you have to observe the color, shape and size of the flower. Yes, you have to identify whether floral whorls like calyx, corolla, androecium and gynoecium are proper there or not. Then you have to note whether the flower is pedicellate or sessile. So pedicellate is what? A flower having stalk. You can see this hibiscus flower have stalk. And sessile means what? The flower which don't have stalk. So this hibiscus flower is pedicellate as it has stalk. You have to also note and observe whether the flower is actinomorphic or zygomorphic. So actinomorphic plants are radial symmetrical that can be bisected into 2 same halves in more than one plane or by any plane. Whereas the zygomorphic plants have bilateral symmetrical flower, that these flowers can be bisected into 2 halves in only one plane. So this Hibiscus rosa sinensis, this is actinomorphic plant, and the flower is actinomorphic as it can be bisected into 2 same half in more than one plane. Then also you have to note whether the flower is unisexual or bisexual. So unisexual is what? Which possesses either stamen or carpel. Here we can see in the hibiscus flower, here stamen and carpel both are present. So this is bisexual flower. So in this way you have to observe certain feature of the flower before dissection. So as this is Hibiscus rosa sinensis or jaswant flower, so it is from the family Malvaceae, and the flower is complete and pedicellate, bractate, actinomorphic, epicalyx. You can see this is what? Epicalyx. So here 5 to 7. This epicalyx, here there are 5 to 7 free green bracteoles are there. So bracteoles are what? The epicalyx is a ring of modified leaves or bract that looks like extra calyx. So all bracteoles, this is what? Bracteoles in a flower, that is called as epicalyx. So single it is bracteole and all together it is called as epicalyx. Now you can observe here the calyx or sepals. They are 5 green, gamosepalous. You can see they are united. Yes, their estivation is velvet estivation. So it has velvet estivation as it is a form of estivation in which sepals and petals are arranged in a whorl and only meet each other's margin. Yes. So this is what velvet estivation. Then if you'll observe this corolla, so here this is what petals, here 5 petals are there. It is arranged in whorl, together is called as corolla, and single it is petals. So you can see here 5 petals, so it is polypetalous, flower is large and showy. You can see red color flower with twisted estivation. As these all 5 petals, they remain free but create an overlapping arrangement at the margin of the appendages. You can see here overlapping. They are overlapping with each other. So this kind of estivation is what called as twisted estivation. Then here this part is what? Androecium. Here you can see at the androecium, here many stamens are there and filaments are fused. So here this is the stamen and you can observe here many stamens are there and filaments are fused. Filaments are fused to form staminal tube and anthers are free here. This yellow part you can see, this is anther. If you will observe the structure of this, it is kidney shape. So this is called as reniform, or you can see kidney shape anther. Gynoecium, this one is gynoecium, you can see. This is gynoecium part. Gynoecium, if you dissect this flower, you will observe this is stigma, this is style, and here inside here ovary is there. It is pentacarpellary syncarpus as 5 carpels are fused. Here style passes through staminal tube. Here at the tip you can see this is stigma 5 and free capitate. So these are the features of this hibiscus flower. Now let's start with the dissection. So for flower dissection, firstly we have to obtain this fresh flower of hibiscus, and then we have to observe different floral whorls in this, like calyx, corolla, androecium and gynoecium. Then we have to take out each floral whorl carefully and you have to arrange them in proper ascending order on a white paper. Yes, so we are going to remove the sepals and then we will place the sepals on this white paper, and similarly we will remove petals and stamens and all, and we are going to arrange all the floral whorls on this white paper. And after the dissection you have to count the members of each whorl. So let's start with the dissection. So first of all I am dissecting this epicalyx. So this is epicalyx. Here the epicalyx is a ring of modified leaves, or we can say bract, that looks like extra calyx. Yes, so this is epicalyx, which are 5 to 7 in number. So here 5 to 7 bracteoles are present here, and altogether it is called as epicalyx. So in this way you have to dissect this epicalyx and arrange them on this white paper. Arrange them carefully, and then I am dissecting this calyx. You can see this green part, this is what calyx, or you can see here in calyx here 5 sepals are there. So dissect like this way. Now you have to dissect it carefully. So this complete whorl is called as calyx. So I am dissecting here this complete whorl. You can see this is what, this is calyx. Yes. So this complete structure is calyx, and if you are separating the single unit, so the single unit is called as sepal. So this single unit is sepal, and altogether the complete whorl, that is calyx. So here 5 sepals, you can see it is green in color, and here you can see velvet estivation. So here you can see velvet estivation as it is arranged in whorl. So now after calyx, we are going to dissect these petals, or in whorl it is called as corolla. So let's dissect this corolla. So very carefully you have to dissect this. Here you can see in corolla you have 5 petals. Yes, you can see 5 petals are there. So we can say it is polypetalous, red color, showy flower, and here you can see in this corolla twisted estivation is seen as these 5 petals remains free, but it create an overlapping arrangement at the margin of the appendages. So here you can see this is the margin, and at the margin of the appendages here you can see this twisted estivation. Yes, as these all petals, they are arranged in overlapping manner. So their estivation is twisted. Now let's dissect these all sepals carefully. You can use forceps also, or you can do with your fingers also. So I am doing like this. You have to do very carefully. Very carefully. These all whorls we have to arrange in proper manner in ascending order. So you can observe here, this is my epicalyx, this is my calyx, and this is my corolla. And here now I am going to dissect the gynoecium part. Here you can observe this is the stigma, yes, and these are the anthers. And on the anther, here you can see these are the anther lobes, yes, which are connected by the connectives, and inside this structure we are going to dissect the gynoecium part. So let's start to dissect the gynoecium part. That you have to do very carefully with the help of needle. So I am using this needle. Just keep it on the paper like this, and with the soft hand just to prick a little, not forcefully, very slowly you have to prick a little, and then straight you have to do like this, very slowly. Yes. Now with the help of your these fingers, easily you can remove this gynoecium. Yes. So you can observe. Here you can observe this is my gynoecium. Yes, which is pentacarpillary, and here its placentation is axile placentation as the style passes through staminal tube. So this is the staminal tube, and here this is the style. So style passes through the staminal tube, and here stigma, you can see here this is the stigma. So here 5 stigma. You can observe in this way, here 5 stigma. So this is our gynoecium. This is the staminal tube, where you can see here many stamens are there. You have to remove one stamen also. Yes. So I am removing here stamen. You can keep it on the white paper. So now in the stamen, you can observe here, this is the yellow part is what? Anther lobe, which is connected to the filament by the connective. So here you can observe this is our stamen. So in this way we are done with the dissection, and now we are going to take a TS of ovary, as this is the ovary, and now we are going to cut the section of it. And for that I am using here blade. You can use razor also. In your dissection box easily you will find razor or blade. So very carefully from here you have to cut half of the ovary, yes, and leave the setup like this only. Then I need one slide. So I am taking one clean slide here. You have to cut a section of this ovary, TS of ovary, very thin structure. So in this way you have to hold like this, and with the help of this blade I am cutting this. So in this way you have to cut the section of ovary. Yes, so this is the TS of ovary. And now take this on this slide, TS of ovary structure. Then this part of this flower again you have to attach here only. This thalamus or half ovary you have to attach here only. Yes. So now you can see this is your TS of ovary. You have to add a drop of water here for the moistening. So in this way we had taken a thin section of TS of ovary with the help of sharp blade, and then you can mount it with a drop of water, and then after that we can observe it under dissecting microscope, and you can count the number of chambers in ovary. Even with the help of this hand lens also easily you can observe the number of chambers in the TS section of ovary. So in this way we have completed with the dissection of flower, and now we'll start with the labeling. So this is my corolla. You have to label it well. This is my corolla. Then this part is my calyx. You have to write here calyx. This is epicalyx. Epicalyx. Then this part is my gynoecium. In the gynoecium you can see this is my here, this is my thalamus. This is style. This is stigma. Yes, 5 stigma here at the tip. Now this is, you can see this is staminal tube. Okay. Yeah, these are the stamen. Here you can see this is my anther lobe. Anther lobe. So this is my anther lobe. This is filament. And here anther lobe is connected with the filament by this point that is called as connective. And this is my transverse section of ovary, that is TS of ovary. So in this way we had done with the flower dissection. Here I had taken hibiscus flower, and marking scheme for this experiment is for 4 marks. This is very compulsory experiment for 4 marks to dissect the flower and display different whorls. So For the display, here 2 marks are there. 1 mark for this TS of ovary. If we'll make the slide of TS of ovary, for this 1 mark is there. For complete dissection and labeling, for this here 2 marks, and you have to make one labeled diagram of this TS of ovary to show number of chambers. For that 1 mark is there. So here, so in your answer sheet, you have to draw the TS of ovary. So you can observe, so very neat and freehand diagram you can draw for TS of ovary. So for that, here you have to draw locules. So here 5 locules. So in this way you have to draw here 5 locules, and in each locule you have to draw 2 ovules which we can observe. So here I had drawn 5 locules which we could see under microscope, and in each locule you can see 2 ovules. So this is my TS of ovary. Here also you should label this, that is locule. Here this part, this is ovule. At the center you can observe this center part, this is placenta. And here each locule is joined with this line, that is septa. So this is my TS of ovary. And here you can see different floral part, or you can write here floral parts of hibiscus. Here we are going to take the transverse section of anther also. For that you have to take another slide. From the other flower, just take anther on this slide with the help of forceps. I am taking anther on my slide, and now with the help of sharp blade, yes, this is my anther, and then with the help of sharp blade, this is the blade, you have to cut a TS of anther. Yes, on the slide only you can cut this anther, and then you have to mount it with a drop of water on the slide, and then you can observe under dissecting microscope. So in this way we had taken the TS of anther and TS of ovary to show number of chambers, and we can easily observe this under dissecting microscope. So you have to dissect your flower in the similar manner, and after dissection you have to display all the floral whorls on a sheet of paper. You have to label it correctly. After making your slide, you can observe under dissecting microscope, and after seeing in the microscope, keep your slides on your paper like this, and you have to note your observation. So in this way, I had dissected the given flower and displayed different whorls and taken the TS of ovary and TS of anther to show number of chambers. Thank you.